So what does the metabolic potential of the community structure differ between the freshwater and marine? So taking that same proxy and kind of expanding it to the global idea. So once again, I can use GC content right off the bat, and I can take 3 replicates from Shark Bay, 3 from Highborn Cave, which is another microbialite forming site in the Bahamas, Pavilion Lake and Kelly Lake. And basically right off the bat, marine and freshwater separate by GC content, just right off the bat. So the microbial community is completely different just based upon GC content. From there, same kind of stamp plot, looking at the functional relationship between freshwater and marine. Basically, the Shark Bay in these kind of purple triangles, the Highborn Cave in these kind of blue circles, Pavilion in kind of these triangles, and then Kelly Lake in squares. And once again, the Pavilion and Kelly are very, very similar to each other versus the marine sites. Looking at the kind of the structure of the community, once again, the Highborn Cave, very, very different. Pavilion and Kelly, very, very similar. So marine and freshwater kind of separate really nicely. Looking at them again with the MetaSeq pipeline through MetaPathway tools, MetaSeq. Of course, you can't probably see all this, but basically, we compare Pavilion and Kelly, Shark Bay, and Highborn Cave. 339 pathways are very, very similar to each other, whether you're marine or freshwater. So that's kind of interesting. And well, of course, Pavilion and Kelly share a lot of similarity to each other, and Shark Bay and Highborn don't have really any real similarities. They're different pathways that are unique to themselves. What's interesting as well is we see that in Shark Bay we have this kind of more ancient kind of carbonate fixation pathway going on in the 4-hydroxyl propionate pathway, and then more photosynthetic genes kind of going on in the Highborn Cave function. So, there. What organisms or organism is responsible for these mini stromatolites within Pavilion Lake? So, what we find that in Pavilion Lake at this one depth, the 20-meter depth, that we find these mini nodular structures that are going on. Of course, this is a kind of freshwater copepod, which in this ecosystem is the shark of the ecosystem. Within there, we have these kind of mini structures, which they have been dubbed nodules. Within these nodules, we just basically pick one of these nodules, and no one really knows what they are. But there was a paper from a group that came before us that basically said that you take these nodular structures and they have mini stromatolite structures that are kind of inside their kind of EPS layer within Pavilion Lake, within these structures. But no one really knew what the heck they were genetically. So I did a quick metagenome and didn't get a lot of data because it's hard to get DNA out of these things. I also did a 16S and found that they are, without a doubt, cyanobacteria. And the closest related sequence is this thing called Calothrix, which is a filamentous nodular forming cyanobacteria. So that's a pretty good sign. As well, within these structures, you see kind of these laminated carbonates basically being precipitated within their cells. So after I did this quick little metagenome, I couldn't really do any more. I couldn't get a full genome, couldn't get any RNA. So I'm basically, well, I really want to get the full story. So you're like, where's the function in all this, Rick? You just showed me a bunch of DNA. You showed me a bunch of stuff about how these things look. Where's the function? Well, I contacted a collaborator and friend, Leonard Foster, and we were able to get lots of protein out of these structures, out of these nodular cyanobacteria. And with the 16S, I've named them as a Candidus phyla because you can't culture it, you have to name it a Candidus. And I've named them Candidus, Calothrix, Pavillionensis, and I've done the proteome. So around 300 predicted proteins in the proteome of the one structure, and of course what structure am I talking about? So you had the green one and you had this kind of pinkish one on the side, and that's the one I'm talking about. So within that, basically I found some hypotheticals, EPS-related carbon fixation, photosynthesis, some phosphorus metabolism, and the list goes on and on. But really what we're after is where is the point where the calcium is actually nucleating? How is the calcium actually nucleating on the structure, and what structural component is actually involved in that? So we have some good hints that it could be this kind of this protein here, a cyanobacterial porin, and it's at high abundance. And of course, this is an abundance of the proteome for each individual peptide. And I basically a little bit of cherry picking here as far as what are the top 2 that are the most abundant and all these different related things. And what was really kind of interesting looking at it is that when I did the proteome, I actually found that there was a phage infection going on. So there's your viral story of the day. There was a phage infection. I found a bunch of tail proteins, peptides, in the proteome, as well as these CRISPR-Cas-associated proteins. And if you don't know what those are, these are basically the bacterial immune system kind of going after these short palindromic repeats against the virus. So one viral story, not much, but there it is. As well as it's green, so you would expect photosynthesis going on. Photosystem 1 and then phycocyanin proteins. Some very interesting kind of weird ones that we're looking at, of course, carbon fixation. You got to have carbon fixation through visco, and then some EPS basically being formed to kind of make these kind of nucleation sites occurring. And of course, some chaperone proteins that are involved in the stress of the cold environment and the phosphorus limitation. So what's the take-home message? We live on a microbial planet. You got anything? Maybe I was going too fast, but that's the main one I want to get today. These are very unique communities, and they've persisted for 85% of our history, and will persist way beyond us and the cockroaches that go whatever. Communities, the microbials appear to have some sort of endemism based on these structural types. So at least with these artichoke structures, we find that they're very similar as far as the microbial community is concerned. And then understanding this vast genetic potential is complex and could be useful for other things. So these things turn CO2, methane, and N2O into rock. So if we figure that out from these guys, maybe we can apply them to other problems that we're dealing with today. And then integration of these genomic and proteomics ideas may obtain more useful information in these uncultivatable organisms. In the case of these unculturable cyanobacteria, I think that once you figure out what it is, the proteome is a way better approach to go after, because the DNA is just impossible to get out. So here's our group. Of course, I was skinnier here. Basically wanted to thank Curtis Suttle for 5 years of looking at these things, and it's been a wild ride, but fun. And my committee, Sue Baldwin in engineering, Tom Beatty and Stephen Hallam. Some folks in the Beatty lab that have been really helpful. The Dipple lab, of course, sitting in the audience, Ian Power and Ann Harrison for my work in the Yukon and northern BC. The Slater lab, where I've done a lot of work on 2 other genomes that I thought I didn't have enough time to talk about today, but it looks like I did. So timing's on. Greg DeVillis, who's helped me with editing various manuscripts, and then all the rest. Thank you very much. I couldn't have done it without a lot of you, and I have a lot of time for questions. Yeah.
So you did a really clear job with the GC plot showing how the freshwater and the marine systems are different. Have you looked at the taxonomic differences between freshwater and marine ecosystems?
So here's the funny part. If you look at the top 5 OTUs, they're all the same. So if you look at marine, it goes absolutely in rank order. It goes alpha proteoid, beta, delta, cyanobacteria, and then the 5th group is kind of one-off. It's either planktonomycetes or nitrospira. But whether you look at marine or freshwater or any of these things, they form these structures. They are always, the top 5 are always the same.
So the top 5 phyla?
Top 5 phyla. Yeah.
And do you, like, maybe, have you guys looked at lower taxonomic levels, like genera?
It's hard to go after the genera. It's really hard, especially with metagenomic data. Even with 16S, it's hard to go after it, because we're talking about very, very short sequences. If you had long 1.5 Sanger-like sequences, then you could get at them. But most of these are obtained through clone libraries still today. Clone libraries at Sanger are still around. It will never die, it seems. And those are where the best phylogenies are done. With 16S, a lot of the next-gen sequencing with 16S is, I don't know, it's kind of all over the place. Because with the 200 base pair sequence, it's very hard to say it's really that organism or not. Cool. Yeah?
What explains the disparity in the number of pathways between the freshwater and the saltwater? So what do you think of that? I found that quite a striking difference.
As far as pathways between.
Yeah, in your pathway analysis, it seemed like you have some shared pathways
Right. But then the freshwater has all these extra pathways that aren't present in salt, the saltwater
Right.
samples, and the saltwater doesn't really have those extra pathways, or their own extra pathways.
A lot of the stuff are basically ubiquitous for the salt, for dealing with salt. So like butane and a lot of the osmotic transporters are basically ubiquitous. So if you'd have them in both things. And so it was kind of striking. As well, the assemblies are not as good with the marine data, and I won't lie and say that they're the same going in, because you're relying on other people's data a lot of the time. So we're hoping with my collaborator at Shark Bay to kind of get more data to see if that improves. But even with trying to surmount more data and looking at the new Illumina data that we just got, basically the same. There's not really anything magical in the marine data sets that are saying, oh, they're completely different. You look at a functional level, they're basically the same. And that goes to the same thing where I look at PI, right? Protein isoelectric point. I think, oh, that'd be all over the place. Not really at all. It's actually really the same, right? Whereas GC just seems to be a better indicator of this kind of fingerprinting, if you will. So hopefully that answered your question.
I was just curious about the EPS, the polysaccharide. Do you have any signatures for it?
As far as?
Do you know what kind of polysaccharide it is? Does that vary from place to place? Because some phages can carry genes for specific types of
It's a really good question.
polysaccharides.
As far as the polysaccharides go, there's not a lot of data, really, as far as what kind of structure the EPS has, and it's really hard to do glycochemistry on these things. We're just now looking at trying to even do glycolipids in some of these structures, and it's just not, the structure's really complex.
So when you say EPS, you picked out certain families of the glycol transferases, for example, or can you stratify it that way?
Right. So when I did a quick KZ analysis of these guys,
yeah.
And so we find lots of weird KZ pathways that are highly represented in some of the sites versus the other ones. But a lot of them aren't like dead ringers, for what type of polysaccharide they can form. But there's definitely, if you look at the EPS-related genes and carbohydrate-specific either enzymes or substrates, they're very different from other systems.
And can you then put them into the different categories as to the asparagus ones or the cabbage ones, or?
I have done that. It's a really good idea, actually. I have not done that, but when I've tried to compare them kind of bulk between different, freshwater and marine, there's not huge things jumping out at me. So, but going after the unique community as a form of structure is really tough.
One over here, one there, one there.
Yeah. So have you ever done, I don't know if it's possible, but have you ever done EM on the different microbialites? Look at the actual structure of the community, maybe there's some sort of biofilm thing going on?
Well, I have. But actually someone sitting right next to you actually helped me out with that. Ian Power actually did some great SEM work on all the different sites that we can find. And with Pavilion and Kelly, we also did these kind of micro sampling where we took samples from the top, bottom sides, because sometimes the structure is like this, sometimes they're like that. And we wanted to know if light had a difference. And they're extremely patchy. I'll say that. They're just kind of, you can't get a very homogeneous, you pick a single point, it's not homogeneous at all. So the way we kind of answer that is we just scrape everything down to where we're getting right into the carbonate and kind of integrate all of that, because they're just so patchy. And even as you go down in depth and structure, they're completely different, right? So that can really add to that kind of moment.
So my question was, in the other freshwater or the marine microbialites, do you see that same lettuce to asparagus to artichoke progression? Or perhaps in the marine ones, do you get like a fennel or a bok choy or something like that? Is it the same kind of depth progression in the...
Well, Pavilion and Kelly are bizarre, and they're the only ones that we found where we have this kind of lettuce patch, cabbage patch thing going on. With Shark Bay, for example, they're basically completely flat, like a piece of bread. They look like basically bread, going back to food analogies. The conal structure, the asparagus kind of conal, that we find the kind of tube-looking structures. We find those in everything from hydromagnesite types of stromatolites in Mexico, silicon-based microbialites and calcite-based microbialites in Yellowstone, and even the hot frying pan lake in New Zealand have these long cone-like structures, which are lake at 80 degrees Celsius. So there's no environmental condition, barring if you have the high alkalinity, you have the high pH and dissolved calcium or magnesium, where these structures can't form, right? You can have them hot, you can have them cold, you can have them salty, they're still going to form. But we don't have this beautiful progression that we see in the building chemical. It's only a BC thing.
I was just wondering, because you're looking at structures at different depths and light and temperature and pressure changes, whether you thought or saw an impact of that on structures.
So light's probably going to be the big one. And getting the data because it's a multi-layered collaboration type of project, so getting the light data to go with all this. We have all the chemical data and trying to integrate. So we get into these highly multi-dimensional things where you have more factors than you actually have replicates to do anything. It's just too many dimensions, so you have to just kind of cherry pick down. My thoughts, light's going to be the one. You don't have light, there's not going to be life, basically, right? So I think that if you were trying to put it into some sort of kind of anecdotal thing, if they have cone structures, I could never prove it, but they have cone structures because they're trying to reach for the light, right? But what's also interesting about light in these structures as well is that they can trap every type of light. They have actually in Shark Bay, they have a new type of they have an unknown cyanobacteria that has this thing called chlorophyll F that can trap the very high infrared light, and so you can go from UV to infrared in these structures. And that would make sense, right? If you're talking about things where life may have began, you have to be able to deal with the carbon sources that you can get and the energy you can obtain. So the abiotic is a big one, and I have a friend that's trying to help me with the mathematical modeling behind that. But once again, it's like 50 factors, not a lot of replication. Even with 3 replicates, it's just not a lot when you have 135 factors. Okay. It's complicated. Yeah.
Yeah, it's just in the initial nucleation of these structures. Do you think it starts with some mineral deposition or some sort of biotic matrix?
So what's interesting is when we were out there in 2011 in Pavilion and Kelly, you see these you can see them in the shallower depths. They basically form on rocks, they form on beer cans, they form on trees, and basically you have this kind of biofilm-ish kind of thing going on, maybe some sort of heterotrophic biofilm, and then cyano gets stuck, can't get out of it, and then they start to filament, and then eventually through time they trap the carbonates around themselves, and then you have this kind of one-off nucleation thing going. So, that would be our best guess, but I don't have time lapse to kind of go through that process because it takes 1,000 years. Start that experiment, and my children's would not be able to finish it. So I would definitely say that.
Sure, go ahead. On that, what about Clinton Lake? You said it formed a lot faster. Is there any research into that?
So with Clinton Creek, so here's an interesting thing about Clinton Creek and Pavilion Lake, which I didn't have enough time to really, well, I guess I did. Lots of time, but basically with Clinton Creek is a very young system, right? So it basically happened in 25 years. And when we look at the sediment communities and the microbialite communities, they're very, very similar, right? So there hasn't been enough time for them to differentiate from each other, right? Whereas in Pavilion Lake, there's been, upwards of 10,000 years. So they have had time to differentiate from each other. So going back to the kind of Mike's question, where you had a biofilm and then you had this kind of nucleation going on, maybe from sediment, from carbonates being rained down from the sediment, from phototrophic alkalization, and then being trapped, or from sediment updrift coming on and then trapping the carbonates in the nucleation point. So we don't see that clear separation. That answers your question.
I'm just curious.
Yeah, they're very weird. The ones in Clinton Creek are weird.
Well, thanks very much, Rick. Thanks.